Welcome back. Now we are going to focus on the procedures on the endosperm culture. So, in procedure, the first and almost thing we want is explant. So, the source of the explant is the most important thing in a endosperm culture. So, in case of cereals, the matured endosperms does not respond to the cultural conditions that we already discussed in above section. That is, in cereals like wheat, barley, and all, there is a fixed time period or an age for the endosperm that will show a response to the culture. So therefore we have to extract at their proper period of development. And normally the endosperms of cereals undergo certain changes after 12 days after the pollination. So this will make them able to respond. So in case of maize, 8 to 11 days after the pollination is considered as a proper period of time for the which will show some response to the endosperm culture. And in case of rice, it is 4 to 7 days after the pollination, and for wheat, it is 8 days after the pollination. And in some families, association of embryo tissues in the initial stage seems to be essential for the proliferation of the matured endosperm. So this is already discussed earlier, that I already said it, that in some cases or in some families, the association of embryo tissue is also essential for their or for the endosperm to undergo the differentiation at their initial stage. And after the initial stage, these embryos can be removed, or these embryos must be removed. And this and then so in this case we can use the entire seed as an explant. And immature seeds provide the explants for the non-endospermic seeds. Next is how explant is inoculated. So we already said this is an in vitro development. So in matured endosperm, the seeds have a massive endosperm tissue. So we have to decode the seed to get the massive endosperm. And the surface is sterilized with a suitable disinfectant and are washing 2-3 times with the sterile distilled water. So here the sterilization is very important. Surface sterilization is very important in endosperm culture. And after this sterilization, this endosperm is planted on a suitable nutrient medium. This is about how the matured endosperm is inoculated. Now next is, in case of immature endosperm, the sometimes the entire seed or the kernel, that is an outside surface, is sterilized and the endosperm tissue is extracted and aseptic condition. This is also similar to the matured one. And in cereals, the top of the kernel or immature ovaries have to be removed with a sterile knife, and exposed endosperm is squeezed out. Here in cereals, we will squeeze the endosperm after cutting off the immature embryos, and after extracting it we can finally place it on the suitable nutrient medium. This is how the immature endosperm is inoculated on it. Now coming to the medias used in the endosperm culture are there are two main or generally used medias which will generally induce the callus. These are basal white media and MS media. So we know in culture media composition can will always be in be the be that factors contains the factors that determines the success growth of triploid plant. So here this medias also contains the factors that helps or generate or induce the callus formation. So Among this basal white and MS medium is the most commonly used basal medium. MS media is the most commonly used one, and this media is supplemented with tomato juice, yeast extract, grape juice, etc. And sucrose is used as a carbohydrate source. And basal media, so basal media is a normal form of a culture media, and this basal media contains sucrose that is used as a source for carbohydrates. And in some cases, along with basal medias, we can add 2,4-Ds or IAA. So IAA is a naturally occurring auxin, and we can also add cytokinin if these are necessary for the growth. And along with basal media, there will be plant growth regulators. These are very essential for the regeneration of the immature endosperm in most species. And the 2,4-D is the most and very important plant growth regulator that have an effective growth or effect that have an effect to induce and that have a high effect to induce the callus formation from the endosperm. So 2,4-D is the most important plant growth regulator that can induce the callus formation. And in some reports, we can say without cytokinin, a successful callus induction has been taken place. But without an auxin, there is no successful callus induction takes place. So without cytokinin, callus can be formed successfully, but auxin is considered as an important for the callus induction. Now next is incubation. Incubation and the maintenance of the culture. To induce the callus, the endosperm cultures are then maintained in the darkness or diffused light. So after the callus formation, we have to maintain this culture. So these cultures are endosperm cultures that are then substituted or then they are placed under darkness or a diffused light, and differentiation takes place when the calli are transferred to a bright light and temperature of 25 degrees Celsius. So this is how we can incubate and maintain the culture. Now next is how the shoot bud is differentiated or embryogenesis takes place in endosperm culture. So plantlet formation, the endosperm culture maturation follows organogenesis or embryogenetic mode of development. So after callus formation, they will either follows organogenesis or embryogenic mode of development. The embryo differentiation occurs when the proliferated tissue is transferred from the callus in basal media with or without gibberellins. So after the callus formation, then it is placed in another media that will help in their differentiation and proliferation of tissue, and that media may be contained or without gibberellins. So culturing carried out up to a complete development, and this culturing carried up to a complete development of triploid plantlet. And the plantlet developed are then subjected to the greenhouse by transferring in vermicompost media and maintaining proper humidity. This is how the shoot bud differentiation or embryogenesis takes place. Now, finally, we are coming into the factors affecting in endosperm culture. So, mainly there are four factors that affects the endosperm culture, that are explant stage or the age of the explant, and next is nutrient medium, and third one is physical factors like temperature, pH, etc. And the fourth one is embryo factors. So let us look one by one. First one is explant stage. That is, we already discussed it about in some cases or in some families there must be a fixed period of time for endosperm to respond to the culture. So that is the same thing is saying here. The proper stage may be vary from cellular to mature endosperm depending upon the species. That that is that is what we already discussed in earlier. The stage or the age of the explant is very important in endosperm culture. And 4-7 DAP in rice, 8 DAP in wheat, 12 DAP in maize are and fully matured in sandalum. These are some examples and not. And next is nutrient medium. This is the second most important thing. A low amount of reduced nitrogen is required for the proliferation, and undefined sources like tomato juice is extract. These are the some so low amount of reduced nitrogen and the tomato juice is extract. These are the some main ingredients or some main uh, factors which is essentially required in a basal medium. The next is physical factors. In physical factors, we know pH, temperature, all this can affect the if all this can forms the physical factors. So the pH of 7 seems to be effective for the fresh weight increase. So pH of 7 seems to be effective, and the temperature, in case of temperature, maximum growth of endosperm occurs between 24 to 27 degrees Celsius. So the maximum growth is between 24 to 27 degrees Celsius and 12 to 16 degree hours photo period, and it requires the light for the embryogenesis and differentiation. It requires a light or a photo period of 12 to 16 hours with diffused daylight. This will support the callus as well as regeneration. So these are the all important things comes under the physical factors. Then last one is embryo factors. That is, association of embryo tissues in initial stage seems to be essential. This is we already discussed in area. That is, sometimes in some cases embryo tissue is also necessary for the proliferation or differentiation of endosperm in the early stages. The same thing is discussing here. And immature seeds of non-endospermic seed exhibits no independence on the embryo factor. So this embryo factor, the association of embryo tissues is essential. It seems to be essential in matured endospermic seed, whereas in immature seeds or immature endosperm culture, the embryo factor does not depends on it, or the as a embryo tissue is not essential in immature endosperm culture.